There are some basic tools that every plant health specialist should have with them when they're going out to handle a field call or conduct a survey looking for an insect or, in our case, a plant pathogen or a plant disease scenario. I guess the basic tools of trade for a plant pathologist would include something like this. A good hand lens, of course, is mandatory. Probably a 10x, some people like a 20x hand lens. The size varies a lot. You have some very small orifices up to these larger size. As we get older, these larger ones are better. But this is a very good way to get a good look at a plant specimen and verify for signs or symptoms of a plant pathogen or disease that we're concerned with. Of course, another good thing you need to have is a nice pocket knife. It can be fairly small, but one that's sharp, in which you can use this to cut cross sections, check roots or other plant parts for other types of symptoms and signs that might be associated with the plant that you're looking at. These days, too, we like to have a camera. Again, you want to record a lot of the problems you're looking at. It might be the field, it might be the plant, it might be a close-up of a leaf or a root that's affected by a problem. Again, capturing that image in its setting in the field and looking at the surrounding settings, let's say looking at the whole field shot to show the perspective that you have maybe a small portion of that field, the center or the edges, that shows some evidence of a problem. It's good to record that again with the camera. Of course, we have digital cameras these days that do a very good job of recording this kind of information. We use a digital camera as well, especially for rapid acquisition and then transferal of that image. The digital cameras work very well. However, for ultimate quality, I still prefer to use a good old 35mm camera or a high quality video camera to acquire images, and then I can use this later for a lot of our research, teaching, and extension work. Another tool besides the camera that has become more important in recent years is a standard GPS unit. Again, global positioning systems unit like this, or there are various handheld ones like this one from Garmin. You'll find quite a variety that will vary in cost from a few $100s to many thousands of dollars, depending upon the accuracy. Something like this is not differentially corrected, but it is accurate to about 25 feet. So again, when you're describing a problem in the field that you're going out to check or surveying, having your road markers, that is road 5 and road XY, 1/4 of a mile this way or that way, east side, north side of the field, but then also having your GPS coordinates and putting that into the official record that you have for that survey will enable you to have more accurate information. You can then go back and acquire more information from other records, from the county, for example, from a grower. And then in our case for pest surveys, this has been very helpful where I can go back next year to an area where we had a disease problem the previous year, and I can look for evidence of overwintering of that pest. I can also look for evidence of spread of a problem from that site to neighboring fields, for example, in that area. So GPS and then using GIS, geographic information systems and mapping, is becoming a more important tool for a lot of us involved with plant health. Of course, if you're taking samples of soil, you'll want to have some kind of a probe that you can utilize if you want to get an idea on the type of soil-borne pathogen populations, for example, that's in a particular field. Something like this where you can attach a plastic bag, you can take a sample to a certain depth, put the soil into the bag, and then you can take the bag, seal it up, identify it, label it, and then put it in a cooler to then transfer back to the laboratory for more processing. There are a variety of ways to take soil samples. Again, if you don't have a probe like this, just a standard shovel, small shovel, or even a large shovel will work, as will a small trowel like this. When we're sampling soil in a field, I like to use a variety of sampling procedures. Some people use what we call the X pattern or the Z pattern or the W pattern. You would kind of overlay that over the field in your mind and walk on a transect and systematically stop maybe every 10 feet, every 20 feet, every 50 feet, and take a soil sample. Place that sample in a bucket, and then you can mix up the soil from that part of the field and then get a representative aliquot out of that to show the amount of pathogen population in the east, the west, the north, the south half of that field, for example. Some people want to take a lot of samples, 20, 30, 40, 50 per field. Some others will take just 1 or 2 samples. So that'll vary depending upon your resources, time, and the problem that you're trying to sample. When you're taking a soil sample, it's important to consider too the depth of the soil to which you will sample. Usually what I try to do is stay within the rooting profile or zone that crop species that I'm surveying or checking will develop. So if I have a shallow rooted crop like onions, for example, that might only develop to 8 to 10 inches of depth, that'll be about the depth I want to sample the soil and get an estimate of the population. If I have a deeper rooted crop species, for example sunflower, that might go down to 2 to 3 feet, then I want to try to get a soil sample from a deeper depth so I know what the distribution of the pathogen will be throughout that rooting depth that plant may encounter during the course of its growth in that season. So just some of the tips, I guess, that we follow for sampling for soil problems. Another trick on sampling, if we have a suspected root health problem in a field, is to try to get as much of the root system and some of the surrounding soil from that affected plant collected and then transferred back to the lab for proper processing and evaluation. You don't want to just pull the plant out of the soil, stripping a lot of the roots and the associated soil, because you might be overlooking a lot of key diagnostic points such as sclerotia of a fungus, might be some insect bodies or parts that are there on the roots that won't come up with that plant is visibly, forcefully extracted from the soil. So again, try to dig up a good amount of soil around that plant and go as deep as you can to get underneath the affected plant part, so you'll have enough tissue and soil to evaluate properly in the field or back in the lab.